electron transport system. So you know very well that after Krebs cycle, the carbon is going to be lost from the acetyl coenzyme via the decarboxylation steps, but due to various dehydrogenation steps during the Krebs cycle, a lot of reduced coenzymes or reduced equivalents will be formed, that is NADH plus H plus and FADH2. These are finally going to participate in ETC, at the end of which there will be a lot of ATP produced. So let's see how it will be happening. First thing, the site where ETC takes place is inner mitochondrial membrane. So to understand where exactly the position or the site where exactly the ETC is happening, I've given the diagram as such. This is inner membrane, this is the intermembrane space, and this is the outer membrane of the mitochondria. As you can see, ETS or ETC consists of 5 complexes. Okay, let us first understand what are these complexes. First complex, complex 1. Okay, complex 1. Complex 1 is called NADH dehydrogenase complex, and it takes up the electrons that are being released by NADH plus H plus when it will be converted back to NAD plus. Okay. Now coming to complex 2. Complex 2 is called succinate dehydrogenase complex. It is going to be allowing the electrons that are going to be released from FADH2. Okay. Now complex 3 is called cytochrome B-C1 complex. Likewise, complex 4 is going to be known as cytochrome C oxidase complex, which consists of also cytochrome A and A3. And finally, we have the complex 5. Complex 5 is called ATP synthetase, or also can be called as the F0, F1 subunit. Okay, oxysomes. Fine. Now, we'll start with complex 1. Complex 1 is NADH dehydrogenase complex. Here you can see that the NADH plus H plus is further being reoxidized to NAD plus, and hence the electrons that are being liberated will be taken up by the complex 1. That is, the first electron acceptor here will be FMN, flavin mononucleotide, and from there the electrons will be passed on to FES, iron sulfur protein. This iron sulfur protein will provide the electrons to a mobile electron acceptor, that is ubiquinone. These are the fixed complexes in the membrane, but ubiquinone and cytochrome C, these are mobile electron carriers. Okay, do remember that. So it will be taken up by a mobile electron carrier. Hence, ubiquinone, by taking the 2 electrons from FES and also by taking 2 protons from the matrix, will finally be converted to ub-hydroquinone, UQH2. Alright. Now, on the other hand, in complex 2 also, the electrons being released from FADH2 will be taken up by iron sulfur protein, and from there it will be finally taken up by another ubiquinone, giving rise to ub-hydroquinone. Okay. Now, main thing that you have to remember here is that when these high energy electrons are being passed through these complexes will use this energy to channelize proton from the matrix. This is the matrix, right? Internal to the inner membrane, it is the matrix. So from the matrix, these particular complexes will now start pumping the protons from the matrix to the intermembrane space. So complex 1 channelizes 4 protons from the matrix. 4 protons are being passed to the intermembrane space by the complex 1. Fine. Complex 1, when it is going to, high energy electron is going to be passed through the complex 1, it will channelize 4 protons into the intermembrane space. The only exception to the ETC, through all the complexes when electrons passes, certain of the protons will be passed, or it will be channelized to intermembrane space, except for complex 2. Complex 2 will not pass on any electron from matrix to the intermembrane space. Only this one being the exception. Rest of the complexes will pass or will channelize protons from the matrix to the intermembrane space when high energy electrons will pass through them. Do you remember this? So now all this UB hydroquinone that are being formed by complex 1 and complex 2 individually, these UB hydroquinone being mobile, they will carry the electrons to complex 3. Fine. In complex 3, the electrons will be first passed on to cytochrome B and then to FES and again from FES to cytochrome C1. Finally, all of these electrons will be channelized to the mobile electron carrier cytochrome C. Cytochrome C will join or will it will transit the electrons to complex 4, okay, that is cytochrome C oxidase complex. So now finally the electrons will reach the last complex, complex 4, and here the 2 electrons will be passed on to cytochrome A, from here to cytochrome A3, and eventually the electron's journey will come to an end when these electrons will combine with 2 protons from the matrix and half of oxygen, giving rise to metabolic water. So metabolic water has been already formed by ETC by the time the electrons reaches the complex 4. Once the complex 4's, the electrons the journey of the electron is completed, in complex 4 metabolic water will be resulted. Okay. So while this electron transport chain was happening, complex 1 will be channelizing 4 protons from the matrix to the intermembrane space. Complex 2 will not do any kind of, it will not be channelizing any proton, that being an exception, I have already told you. Complex 3 will channelize 4 protons as usual like complex 1, and complex 4 will also be channelizing 2 protons. So by the time metabolic water is formed, a lot of proton will be accumulated in the intermembrane space, and the matrix will hence show deficiency of, or it will be lower in its proton concentration, whereas intermembrane space will become high in proton concentration. In short, by the time metabolic water is formed, a proton gradient will be formed between the matrix and between the intermembrane space. Proton concentration becomes high in the matrix, in the intermembrane space, and it becomes low in the matrix. Understood this? So just like every other substance in nature, every substance has to move from higher to lower concentration until an equilibrium is reached, isn't it? So due to the ETC or due to the electron transport system, finally now that a proton gradient has been generated, even the proton would want to move from higher concentration to lower concentration. That means it will have a tendency to now move from intermembrane space to matrix. So this particular thing, or this channelizing the protons from the intermembrane space to matrix is going to be done by the complex 5. That is ATP synthetase. Okay. So every time 3 protons will be passed on through the complex 5, 1 ATP will combine with the inorganic phosphate forming 1 ATP. So this is how by the end of the process, by the end of ETC, a lot of ATP will be produced. Okay, a lot of ATP will be produced. How? When 3 protons are passed through ATP synthetase, 1 ATP will be converted to 1 ATP. Is that clear? Let us now see how many of ATP are being produced by NADH and how many of ATP will be formed due to FADH2. Okay, now see, NADH enters the ETC in complex 1 and the electrons will move through complex 1, complex 3 and complex 4. The electrons that are being released by NADH plus H plus, it is going to run through complex 1, complex 3 and complex 4, isn't it? So total how many protons will be pumped here? 4 plus 4 plus 2, that is 10, right? And in case if we are telling that 3 proton is equal to 1 ATP, so 10 proton is approximately, round off, we can say that 10 proton will be giving rise to 3 ATP. Right? So 1 NADH hence is equivalent to 3 ATP, right? But on the other hand, the FADH2, it enters the ETC only in complex 2. So FADH2 releasing the electrons, these electrons are only going to pass through complex 3 and complex 4, which is going to total proton which are being released here, or the total protons that are being channelized by FADH2 will be only 4 and 2 here. Right? That means 6. So 6 H plus will be equivalent to 2 ATP. Understood that? So that's why 1 NADH is equal to 3 ATP, 1 FADH2 because it enters the ATC later on and it only pumps 6 protons to the intermembrane space. That's why 1 FADH2 is equal to 2 ATP. Understood this? Why it is 3 ATP and why it is 2 ATP? I think all of you are clear about it. So do remember 1 NADH is equivalent to 3 ATP, 1 FADH2 is equivalent to 2 ATP. This will be again required to understand the balance sheet of aerobic respiration. So let's see that.